All right, welcome everyone. You are attending the first session of the AGSx 2025 annual online symposium, which was put together as a way to continue to do presentations in science during the pandemic. And we just carried it on because it's been a great success and a really good venue. Usually your host is Brad Coates. He is busy taking care of grandkids today, so I am stepping in. I'm Brenda Oppert with USDA ARS. And let's just go ahead and get started. I would like to first acknowledge that this is a USDA ARS joint project with I5K. And we have two sessions this year, only two sessions, but they're really good sessions. The first one is today, and you will be hearing about insect genome biology and evolution. Lindsay Perkin will be your moderator. Three really great talks. And session two will be on May 13th, next month. And that one will be on genomic advances in non-model arthropod pests. And Perot, Saleo, and David Lukey at the USDA labs are co-hosting and moderating those talks. And so if you haven't signed up for the session two, feel free to. The link to that is on the right-hand side. I wanted to also acknowledge that there are a lot of people behind the scenes that make this possible. Kevin Hackett is our national program leader and provides a great deal of support and resources, along with Glenn Haynes, who is taking care of today the meeting Zoom and also handles the registration and website. And I told you that Brad is the lead organizer of this symposium. I am going to be your moderator today. And then Anna Childers takes care of the website and Twitter feeds. And Pia Olafsson is also with us today, running the slides and also doing the video recordings. There is a great deal of information resources, which you don't have to grab this right now. I will put this in the chat, but there's information on our website. And then there are other YouTube channels where these talks will be posted, as well as a Slack channel if you want to communicate that way. And so I'm going to turn it over to Lindsay now. I do want to specifically say, and that's what this yellow arrow is in the bottom of your screen, do not post questions in the chat because they may be missed. We are using specifically the Q&A box for those questions. So if you will do that, we'll see those quicker, and the speakers will be online answering your questions as the talks progress. And after the sessions, then they will be answering questions as well. So I'll introduce Lindsay. Stop sharing. So
thank you, Brenda.
Take it away.
Everyone. All right, thanks. Thank you, Brenda. And hello, everyone. Welcome to the webinar today. Like Brenda said, my name's Lindsay Perkin. I'm with ARS, and I'll be moderating the first session on the topic of insect genome biology and evolution. We have a fantastic lineup today. Each one of the speakers is using genomic tools to uncover some interesting biology and evolution in their pest insect system of interest. As we listen to each speaker today, please use the Q&A, as Brenda mentioned, so we don't miss your questions. You can provide questions or feedback, comments, whatever you'd like to share with our speakers today in the Q&A. And then at the end of each presentation, we'll go ahead and read those questions live and let our speakers answer those live for everyone. I'll try to read as many as I can, but depending on time and how many questions, we may not get to all of them live, but the speakers will be answering them in the Q&A as we go as well. Again, Q&A function, we can't stress it enough. We don't want to miss your questions. And it always makes the webinar more interesting when we get questions. So don't be shy and type in whatever's on your mind. We're going to start today with our first speaker, Dr. Hojun Song. He's a professor in entomology at Texas A&M University in College Station, Texas. Dr. Song and his laboratory study insect systematics and evolution on Orthoptera, such as grasshoppers, crickets, and katydids, to understand behavioral, ecological, physiological, morphological, and molecular evolution, all in a phylogenetic framework. So he's going to talk to us today about the evolution of phenotypic plasticity in locusts as inferred from genomic data. We'll go ahead and start that presentation. Enjoy, everyone.
Hello, my name is Hojun Song from Texas A&M University. I'm delighted to be here today to present my talk at the AGSx webinar. The title of my talk is Evolution of Phenotypic Plasticity in Locusts as Inferred from Genomic Data. I'm representing the Behavioral Plasticity Research Institute, which is supported by the National Science Foundation's Biology Integration Institutes program. And then the picture here are the co-authors of my talk. Phenotypic plasticity is defined as the ability of a genotype to express different phenotypes in response to different environmental conditions. This is a ubiquitous feature of life. All organisms on this planet have this ability. We have it, and then many organisms do. Pictured here are some of the most well-known examples of organisms that show dramatic phenotypic plasticity. When studying phenotypic plasticity, it's useful to take what's known as a reaction norm perspective. Reaction norm is a graphical representation of set of phenotypes that can come in response to different environmental conditions, and depending on whatever trait that we're interested, we can quantify by exposing these organisms into different environmental conditions and measure their response. And that could be non-plastic or plastic. Among the organisms that show phenotypic plasticity, I would argue that the locusts represent the most spectacular examples of phenotypic plasticity, which is the subject of today's talk. So what are locusts? Locusts are a special kind of grasshoppers that can actively aggregate and migrate in large numbers, driven by an extreme form of density-dependent phenotypic plasticity. Locusts are familiar to us. They are well recorded in Bible and Quran and other ancient literatures. And they still affect people around the world. Most recently, there is a large upsurge and outbreak of desert locusts from Africa. But they're also found in Central and South America, Australia, and Asia. So they still affect us tremendously. In 2003, Mary Jane West Eberhard published this book called Developmental Plasticity and Evolution. And in this book, she singled out locusts, and locust phase polyphenism, which I'm going to explain, is one of the most striking coordinated alternative phenotypes known in nature. So locusts have this density-dependent phenotypic plasticity, which is known as locust phase polyphenism. At low density, locusts are green or cryptic, and they're shy and they avoid each other. But when favorable environmental conditions arise, the local population density increases, and these locusts will start to change their behavior to be more gregarious, and then eventually become conspicuously colored. This is the two different forms or phases of the same species that's driven by this phenotypic plasticity. I have been studying locusts for many years, and particularly in the genus Schistocerca, which is a grasshopper genus that contains about 50 species. And this is a fantastic group because it contains both locusts and non-locusts. Within this genus, there are three different locust species. Desert locust, Schistocerca gregaria, which is problematic and found in northern Africa and the Middle East. Central American locust, which is found in the Mexico and Central America. And then South American locust, which is found in Argentina and South America. However, the majority of the species in this genus are regular grasshoppers. There are sedentary grasshoppers that do not swarm. Another really interesting thing about this genus is that the majority of the species are found in the New World. But there's only one species that's found in the Old World, which is the infamous desert locust, which is found in Africa. So there's this transatlantic disjunction. Several years ago, I built a phylogeny of Schistocerca based on molecular data and found that the desert locust is at the base of the phylogeny of this genus, and then three locust species do not form a monophyletic group, which suggests that locust phase polyphenism has evolved independently several times within this genus. When we did our divergence time estimate analysis, we found that the genus itself is fairly young, about 8 million years old. And then about that time, there was a aridification of northern Africa that used to be green and foresty that started to dry up, and then expansion of the desert happened. And then that probably created an environmental condition that promoted the evolution of phenotypic plasticity through genetic accommodation. And then possibly around 6 million years ago, a common ancestor of all the New World Schistocerca across the Atlantic Ocean from northern Africa, western northern Africa to South America, and then gave rise to the diversity in the New World through adaptive radiation. So is this possible? Well, in fact, in 1988, there was a large swarm of desert locusts that originated from West Africa that crossed the Atlantic Ocean and landed in Caribbean. If this happened in 1988, it could have happened a long time ago. And so this is a really amazing example of a lineage that came from a common ancestor that crossed the ocean and gave rise to the current diversity in the New World. So based on this phylogeny, we can infer certain things about how the phenotypic plasticity might have evolved within this genus. Because the desert locust is a basal lineage, can infer that the ancestral condition for this genus is the presence of density-dependent phenotypic plasticity, and we'll say in terms of behavior and color. However, when this common ancestor reached the New World, they found themselves in an area where vegetations were abundant. There was really no need to be swarming, no need to keep this phenotypic plasticity, which is known to be an adaptation against heterogeneous environment. So they may have evolved the decreased plasticity or loss of plasticity through genetic assimilation. And then there could be further decrease in genetic plasticity, the evolution of further decrease of plasticity to the point that certain species might have lost completely the ability to respond to change in density. In other words, they evolved the evolution of robustness. It's also possible that certain lineages have re-evolved or gained the increase in plasticity through genetic accommodation again, which may have led to the independent evolution of locust phase polyphenism at least twice. So we're trying to use this as a framework to understand the evolution of phenotypic plasticity within Schistocerca. So study of phenotypic plasticity has been approached in different angles. However, the genetic basis of phenotypic plasticity is still poorly understood. However, when we take the genomic approach, we can think of few different ways how increased or decreased plasticity might have evolved across species. First, it could be due to the differences in genomes. There might be different gene content, different repeats, syntenic patterns, or karyotypes. There are allelic differences between different species that can give rise to differences in phenotypic plasticity. Certain species might have genes that allow them to be more plastic. Or it could be that they might be different in terms of the presence of density responsive genes that express differentially across species. And the way to look at it is by looking at sort of RNA-seq or gene expression level data. It's also possible that there is a differences in epigenomes or methylation pattern or histomodification pattern between the species that differ in their expression of plasticity. So we're not going to talk too much about the epigenomes on this talk, or it could be the combination of above. So the BPRI has focused on the 6 focal species, 3 locust species that have independently evolved an extreme form of phenotypic plasticity, 2 species of grasshoppers that have reduced phenotypic plasticity but still closely related to a locust species, and a single species here that have almost lost their ability to respond to density. So using these 6 species as a model system, in fact, this clade, this Schistocerca as a model clade, we're trying to understand how phenotypic plasticity has evolved across species within this genus. Back in 2009, there was a review paper that was published by Paul Piener and Steve Simpson, who are the leading experts in locust biology. And they said, uncovering the molecular genetics of phase change remains as the final frontier in locust research. However, the progress has been limited by the fact that the genomes has yet to be sequenced for any locust species, a problem which is exacerbated by the huge size of the genomes of grasshoppers and locusts. Yes, so among all insects, grasshoppers and locusts are known to have the largest genome sizes. So it has been very challenging to generate any genomic perspective on the evolution of phenotypic plasticity in locusts. However, thanks to the funding from National Science Foundation and then also funding from USDA and USAID, we were able to sequence the locust genome. So we started with the desert locust genome, most well-known species, taking advantage of a locust swarm that happened in Kenya in 2020. And then we were able to sequence the chromosome level reference genome, which is now integrated into NCBI and I5K workspace. And it's the largest genome of insects that we have sequenced so far, 8.7 gig of genome. Because we were able to figure out how to generate and assemble this high quality, platinum quality genome from the desert locust, we went ahead and sequenced all 6 species of Schistocerca using the combination of PacBio Hi-Fi long reads and then Hi-C. And this is just a summary chart of how much sequencing went in, and then the statistics for the assemblies. And here we have insect assemblies in NCBI, and what we can see is that the quality of our 6 genomes is just off the chart. So we're very, very proud of our achievement. So now we have this genomic perspective, and the 6 species that form the variation in terms of their ability to respond to density as a sort of model clay to really understand the genomic basis of phenotypic plasticity. So first, we were able to sequence high-quality genome and describe their characteristics. We found that the 6 species, the genome is highly synthetic across the 6 species, although there are certain inversions that are evident. These large genomes consist of large repeat regions. In fact, all 6 assemblies are about 70% repeat. And then here what you're seeing is the repeat landscape analysis. Very interestingly, individual species, different species have different patterns in repeat landscape, which means that the age and timing of repetitive bursts varies between species. We were able to use OrthoFinder to identify orthologs across the species, and then we were able to expand this to outgroups. And then all 6 species have a large number of shared orthologs, and then there's large number of one-to-one orthologs. And when we can take this into account and let's ask a question, do species with increased plasticity have more genes than species with decreased plasticity? When we look at the desert locust, the answer might be yes. It has the largest number of protein coding genes. However, this species, Schistoca piscifrons, which is a Central American locust, has a smaller number of protein coding genes, even smaller than non-swarming regular grasshopper. So the immediate answer for this question might be no. As we look a little bit deeper into these genomic data, we found that this large genomes are full of duplications. So there's just tons of gene duplications that happen at the common ancestor of the whole 6 species and each node. And then after species divergence, they accumulate many gene duplication events. And gene duplication events might be important for understanding evolution of certain features. So for example, here we have the Schistoca gregaria desert locust. And then the largest gene expansion in this species is this particular gene NADH ubiquinone oxidoreductase, which might be important for mitochondrial electron transport chain and possibly relevant for swarming. So just by looking at it, maybe there's a correlation between duplication event and gene expression event related to locust biology. However, when we look at non-swarming species, it also has a really large gene expression, which is related to immune function. So in the end, we might think that the species-specific duplication and gene expression events may have nothing to do with locust biology, but has to do with individual species biology. We're collaborating with the folks who developed the Hi-C methods, and Olga and Ares here have developed even more amazing tool to utilize Hi-C data. So based on our 6 genomes, using that as a reference, we're able to inexpensively assemble additional locust genomes using a 3D Carta method. So here's just a diagram of how that approach might happen. So you start with a well assembled reference genome and then sort of low coverage Hi-C data, and then using a series of bioinformatics can actually generate pretty high quality Hi-C assembly of these species with just low coverage Hi-C data. And using that approach, we're able to sequence multiple individuals across several different populations of the Central American locust. And to our surprise, we found that different individuals across populations have a population-specific inversions that are kind of hard to explain. But it seems that numerous population-specific inversions are common within this species. And then also, if you zoom out to species level phylogeny, we're able to see that the numerous species-specific inversions are common across species. Some of these are conserved, some of them have seemed to have evolved independently. So we think that these inversions can potentially explain the genomic landscape and speciation within this genus. Next, another way of thinking about the genomic basis of evolution of increased and decreased plasticity across species is due to the differences in density responsive gene expression patterns. So we're very fortunate to have the facility where we can rear these two species, locust species in different density conditions to induce different reaction norms. And then we call them locust phase time machines. So here we can rear them in high density condition. Here we can then rear them in low density condition to induce different phenotypes. And then having this this rearing facility allow us to track their developmental pattern really closely, and we can manipulate that. So the initial idea was to introduce these 6 species into 2 different density conditions in a standard manner, and then generate transcriptome from head and thorax that are tissue specific and density specific. And then using our genome data as a reference, we were able to identify thousands of genes that are density responsive in the desert locust. So here we're seeing that there are thousands of genes that are upregulated, thousands of genes are downregulated between isolated and crowded condition in thorax and head in the desert locust, which means that there are tons and tons of density responsive genes. When we look at 2 other locust species that show dramatic phenotypic differences between the density conditions, we see hundreds or thousands of genes that are also density dependent, density responsive. However, there's differences across different species. Interestingly, when we look at non-swarming species, we see some spectrum. So Schistosca americana is closely related to Schistosca pisciferans, which does not swarm, but experimentally, they are able to show some level of phenotypic plasticity, and you see that there's a large number of genes that are highly expressed, differentially expressed in this particular species. But these 2 species, Cerealis cubans and nitens, they respond to density very little, meaning that they have a very almost flat or non-plastic reaction norm. And as you can see, the number of genes that are differentially expressed is low, meaning that the number of density responsive differentially expressed genes decrease as phenotypic plasticity decreases. We wanted to see whether there's an overlap among these different species that show phenotypic plasticity, and it turns out that the molecular response to density change appears to be species specific. So here is the heat map, and if there's a consistent pattern across different species, we will expect to see a similar heat map pattern, but each species has a different pattern. When we look at these 3 locust species, still we do not see a pattern that are similar. Instead, we see that there's a species-specific expression pattern across these 3 species, which is quite interesting. Thinking about this pattern, it seems to suggest that these three species has evolved independently evolved machineries that respond to density to express similar phenotypes, but using different mechanisms. However, when we look at their function using gene ontology, these species-specific genes in locusts appear to be functionally similar to each other, which is quite interesting. We look a little bit further into these three locust species and to see if there's any genes that are overlap among hundreds of genes, and then small number of genes, differentially expressed genes, actually do overlap across these three species. So we think that they might be involved in swarming behavior. In the head tissue, there's 32 genes, and then in thorax tissue, there's 55 genes. So what are these genes? Turns out that they are related to things like juvenile hormone, cuticle proteins, and hexamerin, and some other genes that appear to be pretty important. And then they seem to be involved in something that has been informed by other insects that show phenotypic plasticity. For example, in this 4 different species, honeybees, termites, and ants, and horned beetles, some of these genes are known to function as toolkit genes that are involved in phenotypic plasticity. So we seem to be finding that some of these genes that are highly expressed in these locust species might be the same sort of toolkit genes that have been co-opted to be involved in phenotypic plasticity in locust. We have identified some of these genes, and then to validate the function, we have generated a double-stranded RNA, and then we were able to effectively knock down the expression of these genes and then introduce them to different behavior conditions. But really, to understand the transcriptomic response, we generated transcriptome data between GFP control and these target RNAi. And what we found was that knocking out this single gene, these 4 different genes, single gene one at a time, resulted in massive shift in expression pattern. So we are trying to identify what are these genes and then what the function is by doing behavior assay and then further gene annotation. So stay tuned for that one. Finally, we also took this opportunity to look at the time course of solitarization from one density condition to the other, solitarization and gregarization. Because so far the data have been generated from the extreme time points, long-term isolated and long-term crowded. So we were able to rear them in 2 different density conditions and then introduce them to different density for 30-minute, 1 hour, 2 hour, all the way to 72 hours, and dissected specific tissues and then generated data. So for example, when you compare the differential expression pattern of within 72 hours of density shift, we see massive change in metathoracic ganglia, and then the optic lobe. But what's really interesting is that the antenna, there's a lot of genes that are upregulated and downregulated within 72 hours at the periphery of the antennae. So something interesting is going on. It seems that the chemical sensory is really being it's a really important cue for this particular expression of phenotypic plasticity. And then we generated transcriptome data at this different time points. And what we're finding is that within the first 4 hours, there isn't really a whole lot of shift in gene expression pattern. But after about 8 hours, there's a massive shift in gene expression pattern. So these are apparently early acting or upstream genes that are probably involved in phenotypic plasticity, at least in terms of changing behavior, that are expressed from metathoracic ganglia. And we're able to generate the same data going from solitary, so from gregarious individuals to solitary individuals. And we also see the same type of pattern. In the beginning, there's a whole lot of changes, but then later there are massive shifts in expression patterns. So these are important early upstream genes that we need to identify and then try to figure out the function of these genes. So it seems that the genomic basis underlying the evolution of increased and decreased plasticity is not only due to the differences in genomes, but also in differences in transcriptome or expression level. And I'm certain that there's a important epigenetic component as well. So the answer is really the combination of above, and then we are trying to really get at the molecular basis of phenotypic plasticity. So I'd like to end by saying that Schistocerca as a model clade is really ideal for studying phenotypic plasticity, and I hope that more people appreciate this group and then use this as a model system to really understand different aspects of phenotypic plasticity. So with that, I'd like to thank my funding agencies and then the team that helped with this research and then to move this research on locust phase polyphenism forward. Thank you.
All right, thank you, Hojun. That was a fantastic talk. I'm going to start with a couple general questions that came directly to me, and then we'll move to the one you want to talk about live as well. So when you were showing the movement of locusts, potentially from Africa to the New World, I think that's where this question originated. The attendee is asking, how will you predict climate change effect of the American locust species, and do you predict areas of lower moisture or higher temperatures can exert pressure similar to what occurred in Africa?
Sure. That's a great question. So the most recent swarm of the desert locust happened in 2020 in Kenya, which is not an area that is usually prone to locust outbreak, and that was due to very unusual climatic events with just a lot of tropical storms, a lot of rainfall just hitting that area several times continuously, giving rise to really favorable environmental condition, lots of vegetation, and then breeding ground. So that was unusual. Climate change is happening and is making the environment more suitable for certain locust species. Here in North America, we currently do not have, well, here in the US, we currently do not have a locust species. We used to have one called the Rocky Mountain locust, which went extinct apparently. But there is a locust species, Central American locust, which is found in Mexico. And the closest population of locust outbreak of that particular species is in Tamaulipas, which is about 150 miles south of the Texas border. And with collaboration with some USDA APHIS scientists, we were able to do ecological niche modeling to see if the environment in Texas and entire US becomes suitable for the breeding, and all the analysis that we've done makes southern Texas and Florida the totally suitable spot. So, and then we're seeing these Central American locusts possibly moving up. So I wouldn't be surprised if, and do not hope that this happens, but I wouldn't be surprised if we see these swarms kind of moving up north and might be affecting the agriculture in southern Texas.
All right, thank you. Another question is about all those inversions we saw across the different species. Do you think that population-specific inversions occur in other insects?
I've heard that from Fringy, Stephen Roberts, research that it does happen in some Drosophila species. Initially, I was very surprised because when we do genome sequencing, we typically assume that within a species, there's no inversion and everything is sort of stable and fixed. But it turns out that that might not be the case because no one really has the resources to do these sort of high-C type of sequencing across different populations. We don't know the function. We don't know what it does. But apparently that does not affect the biology or survival. So it might be possible if we start looking at different populations using these new tools that can visualize chromosomal arrangement, we might be surprised to see that this pattern might be a bit more general than abnormal.
Yeah, that's interesting. Yeah, we usually think of a species being kind of static. Okay, we're getting some more questions in the chat as well. One you wanted to answer live.
Yeah, well, I didn't actually wanted to answer that live. I just clicked the wrong button,
Okay.
but I
Well,
can talk about that.
Anyway.
Yeah. So the question is, is there evidence in other systems that plasticity is possibly due to the increased number of genes rather than due to function of specific genes? The example that comes to my mind is the aphids, P aphids wing polymorphism, which is also density-dependent phenotypic plasticity. And a recent study from Jen Brisson's group in University of Rochester have shown that there's a particular gene known as a follistatin that has gone through duplication events, and that might be important for regulating wing polyphenism. So there's some evidence that possibly duplications can drive plasticity.
All right, another question is the hope to use results of your studies to help with management of locusts? Example, to use genetic manipulation to inhibit the aggregation of individuals?
Yes, definitely. That'll be sort of the holy grail of what we are trying to do. It'll be amazing if we can manipulate them so that they cannot swarm. The locusts that cannot swarm or they fail to sense the change in density so that we inhibit the expression of these plastic reaction norms. There are challenges. There's like ethical challenges of gene drive. There's actually a challenge of doing CRISPR in locusts. They tend to have a longer developmental period as embryo. So it makes it challenging to integrate them. Large genome makes the CRISPR event much more challenging to do compared to smaller genome insects. So in my lab, we've tried to develop RNAi-based pesticide by feeding them, and because the RNAi works great in locusts. Turns out that locust gut has a lot of double-stranded RNA degrading enzymes that if you feed them, they just degrade them. So we tried using nanoparticle to encapsulate double-stranded RNA, and it's not as simple as one might hope. So yes, there's still hope for doing this sort of genetic manipulation to inhibit aggregation of individuals, and we're working toward that.
Very cool. Yeah, we have the same problems in beetles trying to get RNAi to work via feeding. All right, let's do one more question and then we'll move on. Could you comment on the observed large-scale changes in antennae differentially expressed gene-related gene expression with respect to increased time-dependent competition and pressure of local food resources with dense populations?
Yeah. So this is a important question. So the locust literature so far have really not focused too much on chemical sense, the olfaction. And that's mostly because most of the behavioral work has focused on the importance of tactile stimulus in inducing greggarization and solitarization. But recent studies are showing that pheromones are actually exceptionally important for locust biology. That's for species, within species interaction, sexual interaction. But I imagine that it's also really important for detecting food sources. So this is an untapped area that hasn't really been explored too much, but we have this molecular data that shows that there's this massive shift at the level of olfactory receptors on the periphery of antennae that we didn't really expect. So that's an avenue for further investigation.
Yeah, cool. Yeah, you're doing a lot of cool stuff in your lab. Thank you for presenting today. And I think there's a few more questions in the Q&A if you want to go ahead and type those out. If you come up with a question for Dr. Song while we've already moved on, feel free to add that to the Q&A, but preface it that it's for Dr. Song. All right, so we'll move on to our next speaker, which is Dr. Hunter Walt. He is a postdoctoral associate in the Department of Biochemistry, Nutrition, and Health Promotion at Mississippi State University in Starkville, Mississippi. Dr. Walt uses a variety of omics tools to describe biology of various insects, including bed bugs, black soldier fly, and stink bugs, which is the insect he'll be talking about today. The title of his talk today is Genomic Analysis Sheds Light on a Stink Bug's High Damage to Soybean. Pia, if you'll cue that up, enjoy everyone.
Hello, everyone. My name is Hunter Walt. I'm a postdoctoral associate at Mississippi State University in the Department of Biochemistry, Nutrition, and Health Promotion. First of all, I wanted to thank everyone for being here. And I also wanted to thank Lindsay and the Arthropod Genomics Symposium for allowing me to speak today. And so today I'm going to be talking about some of the work that I did in graduate school, which I recently completed, called Genomic Analysis Sheds Light on a Stink Bug's High Damage to Soybean. So in the first part of this presentation, I'm going to be mainly talking about the red-banded stink bug, which is a major pest of soybean that's present from South America and Uruguay all the way to my backyard in Mississippi in the southeastern United States. It spread across South America with the expansion of soybean production in Brazil and has made its way to being a really big problem for soybean in the southeastern US in the last 20 years. It's consistently designated as one of the most frequently observed stink bugs in soybean, and it's also one of the most damaging stink bugs to soybean. And there's a lot of money that goes into controlling these stink bugs and a lot of money that's lost due to the damage that these stink bugs are doing to soybean. And in general, the stink bug complex is the most devastating pest to soybean. And as you can see on the right here, these stink bugs do quite a number to soybean, rendering their oil and meal value useless. And so I'm specifically interested in the red-banded stink bug because it's known to do more damage to soybean than other stink bugs. And right here, I just have a note from a previous study that was examining just this. They say, note that for Pisadorus gildinii, which is the red-banded stink bug, the protein bodies of soybean are completely dissolved, suggesting the deleterious action of salivary enzymes to seed tissues is greater for this bug compared to that for the other species. And so what we're interested in is looking at the genome of the red-banded stink bug to understand why it has this trait. And so first of all, we formulated a hypothesis or a prediction that the red-banded stink bug does more damage to soybean than these other stink bugs due to lineage-specific expansions of gene families involved in plant tissue digestion. And so what I mean by that is that in the red-banded stink bug's genome specifically, there have been duplications in genes or increases in copy number in genes that are involved in plant tissue digestion. And so to do that, we annotated, or to investigate this, we annotated a publicly available genome for the red-banded stink bug, and then predicted the function of the genes within the genome, assigned these genes to orthologous groups with other hemipteran species. So Hemiptera is the order that stink bugs belong to. And we also use the phylogeny and these orthologs of the Hemiptera to track the gene family evolution across these insects. And that just basically means tracking the duplicated, lost, and gained genes across the Hemipteran phylogeny. And so first of all, as I mentioned, we had to annotate the available red-banded stink bug genome. It's a high-quality genome. We annotated about 13,500 genes in it. We also functionally annotated about 93% of those genes. And then we collected many other species from the order which the red-banded stink bug belongs to, from the Hemiptera. And so right here is the red-banded stink bug. And in this blue box is the rest of the family Pentatomidae, which are other stink bugs. So we have 3 other stink bugs and the red-banded stink bug. And then as you go throughout the tree, these are lineages that are more distantly related to the red-banded stink bug. In the right over here, we just have some statistics about our orthology assignments. And so we see there are many genes that are present in most of the taxa, but there are also some genes that are specific to the Pentatomidae and some genes that are even specific to lineages. And so, as I mentioned earlier, we're able to track the duplicated genes across the phylogeny. And so we are specifically interested in the red-banded stink bug lineage. And so we were able to obtain the set of duplicated genes within the red-banded stink bug lineage. And we found that, consistent with our hypothesis, this set of duplicated genes is enriched with functions that are involved in digestion, such as proteolysis. So as you can see, there are many genes that are serine proteases, trypsins, ovochimases, and cathepsins. And so this is, as I mentioned, consistent with our hypothesis that the red-banded stink bug does in fact have expansions of digestion-related gene families. What's also interesting here, that's a little bit off the subject of this study, or a little bit something that we're not as interested in for this study, but is still interesting overall, is that we also see genes that are involved in detoxification that are expanded in the red-banded stink bug lineage. And these genes can also be used to detoxify pesticides. And the red-banded stink bug is notoriously hard to control. But for this study, we were mainly interested in its digestion trait. Okay, so we were happy to see that our first prediction was supported. So now we made another hypothesis. We predict that the expanded digestion gene families are highly expressed in the red-banded stink bug's salivary tissues. Okay, and this is because the way that stink bugs and other hemipteran insects feed is through their piercing, sucking mouth parts. It's characteristic of the order. And so basically what they do is they take these straw-like mouth parts that they stab into a plant and eject a vile concoction of salivary enzymes that digest much of their food outside of the mouth parts and outside of, or actually in the plant. And then they slurp it back up through their straw-like mouth parts. And so we think that much of the digestion-related genes that are expanded would be differentially expressed in the red-banded stink bug lineage, which could explain its higher damage to soybean compared to other stink bugs. And so to do this, we had to crack open a lot of stink bugs and dissect their salivary tissues. And the red-banded or stink bugs have three distinct salivary tissues. There's the principal salivary gland, which you'll see denoted as the PSG through a lot of this talk. There's the accessory salivary gland, which you'll see denoted as the ASG through a lot of this talk. And then those are connected by the accessory salivary ducts. And so we took all of these distinct tissues and subjected them to RNA-seq, while we also sequenced the rest of the stink bug bodies without the salivary tissues so that we could compare the salivary tissues to the rest of the tissues without the salivary tissues so that we could understand what genes are differentially expressed in the salivary glands compared to the rest of the body. And so here it is. We have the salivary tissues versus the rest of the body without the head. And so for this study, I'm mainly going to be focused on the principal salivary gland because that's the salivary tissue where most of the digestive enzymes are made. And so we conducted a differential expression analysis, and what you can see is going up this y-axis. These are the genes that are differentially expressed in the principal salivary gland of the red-banded stink bug versus the rest of the body. And so we also overlaid the duplicated and gain genes on these on this chart. And as you can see, there are quite a few duplicated genes right here in red that are differentially expressed in the salivary tissues, but it's really no more than you would expect by chance. And so we were able to run simulations to see how many duplicated and differentially expressed genes you would expect at random. And what we see is really no different than that. And so this is contrary to our hypothesis. We predicted that these expanded gene families would be differentially expressed in the salivary tissues, but there's really no more duplicated and differentially expressed genes than you would expect at random. And so we started to think about different approaches, or other hypotheses or routes we could look into. And we thought that maybe the higher damage of the red-banded stink bug could be due to differential retention instead of differential expression, or some kind of genomic innovation where duplicated genes are driving the digestive capability. And so what I mean by differential retention are the genes that were kept in the red-banded stink bug lineage, but were lost in these other stink bug lineages. So as I mentioned earlier, in our gene family evolution analysis, we can actually see what genes were lost in other stink bug lineages relative to the red-banded stink bug. And so we came up with these plots right here where we have, so I know it's a lot to look at, but in each of those plots are the same genes. Those are the differentially expressed genes in the red-banded stink bug lineage. Okay, all of the dots are those genes, but we overlaid genes that were lost in these other stink bug lineages, the brown marmorated stink bug, the southern green stink bug, and the neotropical brown stink bug in red. And so what we see right off the bat is there's quite a few of those of genes that are differentially expressed in the red-banded stink bug salivary gland that were lost in these other stink bug lineages. And when you look at what functions are enriched in these set of differentially retained genes, you see that they are enriched with more of these digestive genes, so genes involved in proteolysis and catabolic processes. And so we think that this may explain the red-banded stink bugs higher damage to soybean more so than the genomic innovations or duplications. And so this also makes sense that these proteolysis genes are enriched because stink bugs, red-banded stink bugs primarily feed on legumes, which are a protein-rich plant. And so this is why we think that these genes may have been retained in the red-banded stink bug that were lost in these other lineages. And so this is another excerpt from a paper that basically says that red-banded stink bugs are really only ever observed on legumes. The only time they're not is if soybeans aren't around, while other stink bugs can be seen in other crops and even on fruits and things like that. So they're more generalist feeders, while the red-banded stink bug is basically a legume specialist. They only really feed on plants that are in the family that legumes belong to. And so what we think is that this pressured these proteolytic genes to stay in the red-banded stink bug while they were less important in the other stink bug species, and they may have randomly lost some of those genes that are now no longer expressed, or sorry, they now no longer have, but are differentially expressed still in the red-banded stink bug salivary tissues. And so our conclusions here is that expansions of gene families in the red-banded stink bug lineage do not explain its salivary digestive capability compared to other stink bugs, but differential retention is a more appropriate way to explain this. And this makes sense. This is how I bet many things happen, where you're retaining functions instead of creating new functions, which is a lot more difficult. So this study was all transcriptomic, though, so we would need more confirmation using proteomic studies and even functional studies of some of these genes that are present in the red-banded stink bugs' salivary tissues. So while we were going through our analysis, our differential expression analysis, we noticed multiple genes that were differentially expressed in the red-banded stink bug salivary gland. So this is that same chart that I showed you earlier of differential expression. These are the genes that are differentially expressed in the red-banded stink bug principal salivary gland. But now we have these bacterial type genes that are upregulated inside of the red-banded stink bug principal salivary gland. And so we were interested in these genes because they're glycoside hydrolases. And glycoside hydrolases are proteins that break down complex carbohydrates into simple sugars, and many times are used in plant tissue metabolism. So naturally, we were interested in these genes, but we were skeptical as to if these were artifacts or not, because whenever we basically blasted these genes against databases, they hit bacterial genes and not a bunch of other insect genes. So we were worried that this may be due to bacterial contamination. So we checked if this was the case, and it's likely not the case. And that's because these genes have eukaryotic properties. So they have a short intron, which most prokaryotic genes do not have. And they also have a eukaryotic type signal peptide. The domain that's encoded in this gene is a glycoside hydrolase family 26, which is present in bacteria and fungi, but it's really never been seen in insects before. So we wanted to see if these genes were actually present in other insects, but and we found them, but that's really restricted to two closely related hemipteran superfamilies. And so these genes are present in the Lygoidea, the superfamily that that includes seed bugs, and also the Pentatomoidea, which is the superfamily that includes stink bugs and shield bugs and their relatives. And so, like I said, it's only present in these two closely related superfamilies. It's present in a high copy number or many hits in the databases that we screened. And I have some species in bold in this table because those are the species that we use for the rest of the analysis, which are the species with actual annotations. So we moved forward with these to do some more analysis. And so I'm not sure if you could pick up on what I was hinting at, but we were wondering if this gene was actually acquired by stink bugs via a horizontal gene transfer event. And so what a horizontal gene transfer event is when a gene is shared between two organisms, but it bypasses the need for sexual reproduction. Now, this happens all the time between bacteria. as they are sharing genes between each other, but this doesn't always happen in eukaryotes. It's a lot more rare. And so we took all of the insect GH26s that were annotated in these genomes, and we took a selection of bacterial and fungal GH26s and put them in a tree together. And so you can see right here are the insect GH26s, and green are the bacterial GH26s, and in that gold color are the fungal GH26s. And so now taking a closer look at the tree, we have a clade down here that's fungi and bacteria. And we have a clade right here that's insects and bacteria, which share a most recent common ancestor. And so this is indicative of a horizontal gene transfer event that this bacterial clade and the insect clade shares a most recent common ancestor with high support. If this weren't a horizontal gene transfer event, you would expect this tree to look more like the tree of life, with the fungal GH26s or the other eukaryotic GH26s being sister to the insects. But what we see is that this clade of bacteria share a more recent common ancestor with the insect GH26s, indicating that this was a horizontal gene transfer event that happened at the common ancestor of seed bugs and stink bugs. And so here's just an overview. So we took the topology of a tree from a previous paper of the infraorder that many of these species belong to. And so this horizontal gene transfer event had to have happened around 217 million years ago for it to have been at the common ancestor of the Lygoidea, the superfamily including the seed bugs, and the Pentatomoidea, the superfamily including the stink bugs. But interestingly, this was lost twice independently in between. And so, like I said, we're following a topology that was from a previous paper. So it could be actually different topology, but nonetheless, there's also not a ton of genomic information about these two superfamilies. So maybe that's why we're not picking up this horizontally transferred gene in these lineages, but they may actually have been lost in these lineages. And so what's interesting now is taking a closer look at these GH26s, which you saw maybe earlier how many hits we were getting against the insect genomes in that table I showed earlier. This gene was dramatically expanded in insects. And so this GH26 gene is this pink color. And as you can see, there are just a ton of them whenever they're present. And so in stink bugs, we have some more contiguous genome assemblies. And we see that these GH26s are typically flanked by the same gene, which indicates that it was a single horizontal transfer event. But nonetheless, it's remarkable how much these genes have expanded, which indicates that they're likely beneficial to have in the genome. Otherwise, if it were to duplicate and not have been beneficial, it would have been purged from the genome fairly quickly. And also, if it were not beneficial, if the horizontal gene transfer event were not beneficial in the first place, then it likely would have been purged from the genome. And so next, we wanted to see if these GH26s were differentially expressed in the salivary tissues of other stink bugs, or really any other insect that we could find it in with tissue level data sets. And what we found is just that the stink bug GH26s are biased in their expression towards the salivary tissues. And I tried to make this easy to digest. All of the bars in shades of red are salivary tissues. The bars in shades of blue are tissues that could contain, are data sets that could contain gut tissues, which are another site of digestion in insects. And then the other tissues are in gray. And what we see is overwhelmingly these GH26s are biased towards the salivary tissues, specifically to the principal salivary gland when information is available about the principal salivary gland. And so this indicates that this gene is likely involved in feeding. As I mentioned earlier, it has a signal peptide, so it's likely secreted into the saliva and could be playing a role in breaking down some of the complex carbohydrates inside of plant tissues. And so, as I mentioned just then, that's exactly what these genes do, glycoside hydrolases. They break down complex carbohydrates into simple sugars. And they're important metabolizers in many plant cellular processes because plant cell walls have a lot of these sugars incorporated into them, specifically glycoside hydrolase family 26, which is the predicted glycoside hydrolase domain that's in the stink bug lineages, is an endo-1,4 beta monosidase. And so its substrate that it acts on are mannan sugars and galactomannan sugars. And so what's pretty interesting here is that there have been previous studies of bacterial and fungal GH26s. I don't know if you noticed in my tree, I had stars next to GH26s that have been functionally validated. But some of these studies, or most of these studies, have found that these GH26s are breaking down sugars that are enriched in legume seed endosperm. So as you can see right here, this GH26 from a bacteria has high activity towards locust bean gum, which comes from a legume, konjac powder, which I believe comes from a legume, and other mannan sugars. But along, when you look at the other carbohydrates they tested, it doesn't really have activity towards those. And so this is interesting to me because it has agricultural relevance, because stink bugs are a major soybean pest, which is our most valuable legume. And so this GH26 may have informed stink bugs' success in soybean because they are able to break down these sugars that are highly enriched in legume seed tissues, including soybean. And so my conclusions here are that insect GH26s were horizontally transferred from bacteria around 217 million years ago. These genes then assimilated to eukaryotic biology by gaining introns and eukaryotic signal peptides. And then very interestingly, they were dramatically expanded in stink bugs and seed bugs, but were lost in other lineages. And this is likely associated with feeding habits. And so we would expect these genes to be in higher copy number in species that feed more on plants that have the specific sugar substrate that GH26s act upon. So as I mentioned earlier, legumes, which stink bugs feed on, a lot of stink bugs feed on. Also, we noticed that these genes were not present in predatory stink bugs. And so this further corroborates our observation that these genes are likely involved in plant feeding because the predatory stink bugs that eat other bugs don't have them in their genome anymore, likely because they didn't need them to break down the sugars that are present in other insects. These GH26s also show biased expression to the principal salivary glands, which indicates their role in feeding, because as I mentioned earlier, the principal salivary gland is the site of where most of these digestive enzymes that go into the saliva are made. And then these GH26s may be critical to stink bugs' success as soybean pests because it may be helping them break down some of the complex carbohydrates that are enriched in the seed pods, which, as you saw earlier, the stink bugs will just do a lot of damage to them. And so with that, I want to acknowledge our funder who helped us pay for some of the sequencing here, and my collaborators and co-authors on the papers. There's linked to the papers that came out from these. And with that, I will take any questions that you have.
All right, thank you, Hunter. That was a fantastic talk. Really cool biology happening there. We have several questions. Some people are putting their questions in the chat. I just want to remind you to put that in the Q&A so that our speakers can see them. But I have a couple that I'll read out from the chat right now. First, I have a question. So I'm starting to work in a system that we think we see horizontal gene transfer. And when we kind of zoom out and look at the areas around that event, we think we're seeing some sort of pattern. Have you looked at that, and have you noticed if there's certain genome features around the horizontally transferred genes that give you clues to how they're inserted perhaps, or something like that?
Well, as far as genomic features go, structural features, not as much, but when we looked at our horizontal gene transfer event, we looked at the synteny and the flanking genes around it, basically to confirm that it was one horizontal gene transfer event versus multiple horizontal gene transfer events. But as far as like signature sequences that are flanking the horizontal gene, the horizontally transferred gene itself, we didn't identify anything consistent across those genomes, because we were mainly just looking to see if the same genes were flanking the horizontally transferred gene in all of the insects that we had genomes for. So,
okay, yeah, very cool. We're not sure what we're seeing, but we think there might be like a lot of repeats, and we don't know if the repeat area was just like a good place for those to be inserted, or if they happened after the insertion due to replication. So anyway, maybe we'll have to check notes with each other.
right.
All right, another question from the chat, and this is from Brenda, and you can unmute, Brenda, if you want to ask it yourself. I'm also interested because it has to do with the project I worked on when I was in her lab. And that is, have you tried knocking down some of these digestive genes, or proteolytic genes, and seeing what happens?
So that is the plan. We haven't done that yet because we're a mainly computational lab. And so we are applying for funding to do those types of things, and looking for collaborators actually to do those types of things. So we'd love to work with anybody who'd be interested in helping us with that, but we have not actually tried to knock any of them out. So
Yeah, I think that Lindsay would be a good collaborator in respect. Lindsay, Wayne Hunter has a question, and I've unmuted him, so I don't know if you can see him, but Wayne, do you want to ask a question?
Can you hear me?
Yeah, we can.
Hey, Brenda, Lindsay, it's good to see you both. Yeah, so on the stink bugs, I was wondering if they sorted out, the stink bugs, the way your evolutionary tree sorts out is because it corresponds with evolutionary development of seeds and flowering plants, and they became seed feeders, and seeds are high, very solid. But stink bugs also belong and feed on plant tissue from time to time, and there should be watery saliva enzymes and proteins. And I was curious if you tried to hunt down by association to hemipterans the watery saliva that would be linked to liquid feeding diets.
So that's a good question. I will say that for this talk, we mainly focused on the principal salivary gland, which is the site of where most of those watery digestive, or the digestive enzymes in the watery saliva are held. I will say that there are, when you blast the genes that are differentially expressed in the red-banded stink bug salivary tissues, they are highly similar to other stink bug species that may feed on not necessarily seed tissues, but like sap and things like that in plants. But we were specifically interested in the red-banded stink bug's trait of being able to digest those seed tissues. So that was our main focus.
Okay, thank you. Yeah, good question, Wayne. Thanks for speaking up. We have one more question in the Q&A. Do stink bugs have any specialized organ or tissue reservoirs that house bacterial endosymbionts?
So that's a great question. Yes, stink bugs many times are associated with bacterial endosymbionts, and they live a lot of times in these crypts within the gut, so these folds within the gut of the stink bug. And so I will point out in that tree that I made of the horizontal gene transfer event, in the clade, the bacterial clade that was sister to the insect GH26s, there were some bacteria that are associated frequently with stink bugs like Pantoa or Pantoia, I hope I'm pronouncing it correctly, that often form these endosymbiont relationships with stink bugs. But it's hard to tell because the horizontal gene transfer event happened so long ago if it happened to be derived from some endosymbiont within the stink bug. But yes, stink bugs do oftentimes associate with endosymbionts.
Cool. Yeah, thank you. And I think at this time, we're going to move to the next speaker. So thank you so much, Hunter. And if you think of a question for him, just like I said before, make sure you preface in the Q&A that it's for Dr. Walt.
Thank y'all.
Okay. Yeah, thank you so much. All right. Our final speaker today is Dr. Megan Fritz. She's a professor in the Department of Entomology at the University of Maryland in College Park. Dr. Fritz studies arthropod evolution in response to human-mediated environmental change. She and her lab use molecular, genomic, and computational tools to shed light on the genomic variants that facilitate adaptation. Her talk today is entitled Genome Evolution in a North American Lepidopteran Species in Response to Modern Agricultural Practices.
Hello, my name is Megan Fritz, and I'm an associate professor in the Department of Entomology at the University of Maryland. Today, I'm excited to have the opportunity to share my lab's work on genome evolution in a North American Lepidopteran species in response to modern agricultural practices. In my lab, we study the ways in which humans interact with the natural world, and in doing so, shape communities of species within it. These interactions include sculpting the physical terrain, altering the biotic composition of species in an ecosystem, or through chemical inputs into the environment. We're interested in how insect management approaches, which represent major human-imposed environmental inputs, shape the genome architecture of exposed populations. The management approaches we've considered include synthetic, sprayable pesticides, as well as transgenic crops that express plant-incorporated pesticidal toxins. These inputs into the environment place extraordinary selection pressure on arthropods to evolve resistance to their toxic effects. This figure shows the trajectory of arthropod resistance evolution from 1914 through 2022. As of 2022, 625 arthropod species were classified as resistant to at least 360 pesticidal technologies. The genomic architecture of resistance has long been thought to be driven by the strength and nature of pesticide selection. Of course, selection is often pesticide-specific. Gene targets of selection depend upon the pesticide mode of action and molecular mechanisms underlying toxicity. But the strength of selection was also thought to determine the number and types of mutations involved in pesticide resistance. As an example, this figure from a review by Mackenzie and Batterham shows the distribution of pesticide resistance in a population of insects prior to selection with one of two different selection strengths. A represents a pesticidal concentration that kills much of the population, but a small proportion of individuals systematically survive. In other words, pests selected by A receive a relatively low dose. B represents a high dose of pesticides, a concentration much higher than what's required to kill all individuals in the original population. After many generations of selection, pest populations that have experienced different selection strengths show shifts in phenotype or degree of resistance as represented by these hatched frequency distributions. Mackenzie and Batterham and others before them predicted that, in general, pest populations experiencing selection by low doses of pesticide, or A, would experience a shift in resistance phenotype that was driven by many changes of small effect size at multiple loci throughout the genome, meaning that resistance would have a polygenic architecture. However, pest populations experiencing high doses of pesticide, or B, would likely require one mutation of very large effect size, usually a loss of function mutation in a gene, rendering a pesticide receptor unable to bind the pesticide. For many years, most pesticide resistance work in wild pests focused on identifying the latter case, a monogenic resistance architecture, partly due to limitations of sequencing technology. Empirical evidence to support a polygenic resistance architecture in wild arthropod populations has been limited, giving the impression that most field-evolved resistance has a monogenic architecture. I began my lab at the University of Maryland in 2016. This is a year when Helicobacter picea, a major pest of corn and cotton in North America, evolved high levels of resistance to Bt transgenic crops. Bt crops comprise nearly 90% of corn and cotton acreage, and strong adoption of this technology by growers since its commercialization in 1996 resulted in very strong selection pressure to overcome the effects of their transgenic insecticidal proteins. Bt crops have been genetically engineered to express plant protective transgenes. These transgenes are derived from the bacterium Bacillus thuringiensis and encode crystalline proteins expressed throughout plant tissues. I'll refer to these proteins as cryotoxins throughout the rest of the talk. Plants can express a single toxin or multiple cryotoxins together in their tissues for their protection from insect feeding. Insect ingestion of transgenic plant tissues exposes them to cryotoxins. Due to a series of biochemical interactions between the toxins and receptors in the insect midgut, the result of toxin ingestion is typically stunted growth and mortality. It had been predicted that Zea would evolve resistance to Bt crops because Bt crops do not produce a high enough dose of toxins relative to what's required to kill susceptible individuals, and that includes resistant heterozygotes. Zea had low susceptibility to cryotoxins even prior to their commercial release in Bt crops. This provided an opportunity to empirically test the theoretical prediction that low-dose exposure results in a polygenic resistance architecture, even in field settings. We use this system to ask the following questions. What is the genomic architecture of resistance to transgenic Bt crops in Helicobacter picea? And we hypothesized that given their low dose exposure to cryotoxins, resistance would have a polygenic trait architecture. We also asked, what are the types of mutations that allowed for their rapid evolutionary response? So for the rest of our time, I'm going to present work by two former members of my lab, Dr. Kate Taylor, who was a postdoc at the time, and her undergraduate assistant, Jane Quackenbush. And this work was recently published in the journal BMC Genomics. So to address this first question, we conducted a replicated quantitative trait locus or QTL analysis using recently collected field-resistant and laboratory-reared susceptible Helicobacter picea as cross-founders. For our QTL analysis, we quantified resistance using a diet incorporation bioassay. Over 800 F2 progeny from 10 different crosses were placed onto a diet for which lyophilized corn leaf tissue had been incorporated. These progeny were grown for 7 days and then weighed. Susceptible insects should not grow on leaf tissue containing toxin, whereas resistant individuals can, and therefore weight was our metric of resistance. We assayed larval growth for 2 different treatment comparisons for each of 2 toxin types. In our comparisons, half of the F2 progeny from each family were grown on our treatment of interest, diet containing tissue from a toxin-expressing corn isoline. However, we also examined growth on the same corn near isoline, but without toxin as a control. This allowed us to separate resistance-related growth from growth in general. Now, the architectures of these two traits did not overlap, and therefore, I'm not going to talk any further about our control treatments today, but I would invite you to look at our publication if you're interested. After the weights of assayed F2s were recorded, we extracted their DNA along with DNA from the cross-founding grandparents and sequenced their genomes. Grandparents were sequenced by whole genome resequencing, and F2s by GBS, or genotyping by sequencing. So here are the results of our QTL analysis. On the x-axis, you have chromosome number, and on the y-axis, you have smooth additive effects of having one resistant parent allele on larval weight gain in milligrams. The points in red represent regions that were statistically significantly associated with growth on each toxin. The top panel shows QTL for Cry1AB resistance, and the bottom panel shows QTL for resistance to leaf tissue expressing two toxins, Cry1A.105 and Cry2AB2. If we focus on the top panel, we can see that there are at least 5 chromosomes associated with Zea resistance to Cry1AB. And this has historically been considered a low-dose toxin for Zea. These chromosomes were 2, 3, 9, 21, and 30, showing that Cry1AB resistance in field-collected Zea has multiple underlying genomic regions and a polygenic trait architecture. Resistance to Cry1A.105 and Cry2AB2, on the other hand, seemed to localize to chromosome 9 and likely involved multiple regions of this chromosome due to the elevated additive effects across the entire chromosome. So for the rest of my talk, I'm going to primarily focus on Cry1AB resistance. So to address the second question, what are the types of mutations that allowed for their rapid evolutionary response? We paired the results from our replicated QTL analysis with multiple additional experiments. The first was a differential gene expression analysis. Together, our QTL experiment and differential gene expression analysis tested whether QTL regions contain genes that were strongly differentially expressed between resistant and susceptible Zea. This provides insight into genes or gene regions potentially involved in resistance. Wild Zea were collected from either BT expressing or non-expressing corn, and we used a susceptible lab-reared population grown on untreated diet as a control. Because cry-toxin activity takes place in the insect midgut, we dissected midguts from larvae of each of these populations, pooling 4 midguts per replicate with 8 replicates per population. RNA was isolated from these midgut pools, then prepared into libraries and sequenced by Illumina. We analyzed our data using a standard differential gene expression analysis, where gene expression levels were compared between populations. Now, recall from our QTL analysis that chromosome 9 had the strongest effect of all loci on Cry1AB resistance in Helicoverpa zea. Here, our top panel zooms in on our chromosome 9 QTL peak. The bottom panel shows our differential gene expression analysis data for genes across chromosome 9. The y-axis shows gene upregulation as the log fold change in the field-collected resistant samples relative to laboratory-reared susceptible samples. Notice that in the peak of our QTL, we identified a cluster of genes that were all upregulated in resistant field-collected samples. So our strongest effect QTL region contained a cluster of differentially expressed genes in the larval midgut. It's unusual for an entire cluster of genes to be simultaneously upregulated, And we reasoned that this pattern of upregulation may be due to a gene amplification event. An increase in the number of copies of a gene in the genome could result in the appearance of increased gene expression. To test this, we conducted a read depth analysis. Whole genome sequencing reads from grandparental populations used in our crosses were mapped back to the reference genome, and the depth of coverage across chromosome 9 was examined. So how does this work? Imagine a reference genome that looked like the genome in blue. This is the condition for susceptible individuals, where each susceptible haplotype contains a single copy of each target region. Now imagine haplotypes from resistant individuals in beige contain an extra copy of that target region D. When we map whole genome sequencing reads of those resistant individuals back to the reference genome, most reads pile up to some expected depth, which should be within some window of variation around the depth of coverage you aimed to sequence each sample at in your whole genome sequencing experiment. But for individuals that have an extra copy of the target region, their read depth is higher. That's because the extra sequencing reads map back to the single copy reference genome in the same place reads from the original copy does. So a gene amplification event associated with resistance should result in higher read depth in amplified regions. So to test for increased copy numbers of the gene in our QTL region, we examined the read depth across chromosome 9 in resistant grandparents from our QTL analysis. Here in this figure, X represents the position on chromosome 9, and here we're narrowed in on the region containing strongly differentially expressed genes between resistant and susceptible populations. Y represents the average relative coverage depth of this region compared to the average read depth across the rest of the chromosome. Points represent the relative depth of a 10 kb region for a single individual at a single chromosomal position. Relative to the rest of the chromosome, the region showing simultaneously upregulation of gene expression had higher depth of coverage, and in some cases, up to 4 times higher in resistant individuals. So when results of these two experiments were combined, increased gene expression and coverage depth in resistant Helicobacter suggested a potential gene amplification event was likely the mutation underlying resistance in our chromosome 9 QTL region. We also compared read depth across chromosome 9 in both susceptible and resistant cross-founding grandparents. While we see no variation in read depth in susceptible individuals, that's the panel on the far left, this indicates that they're a single copy in the region. On the other hand, in the middle panel, we see an increase in read depth in resistant Zea from the field. Finally, to confirm copy number variation in this region, we used droplet digital PCR. This is the gold standard in detection of copy number variation. And for this, we targeted one gene sort of in the middle of this cluster of genes, and the gene was annotated as trypsin 77. The far right plot shows the correlation between mean copy number detected using coverage depth as compared to copy number estimated by droplet digital PCR. The gray points represent the susceptible cross-founding parents, and ddPCR confirmed they were all single copy. Field-collected resistant cross-founders, shown in color, were all at least homozygous for 2 copies of this gene cluster. Finally, to demonstrate the field relevance of this mutation, we took a temporal genome scanning approach. We systematically sampled Zea by pheromone-baited trap from a major BT corn and cotton growing area of Louisiana, starting in 2002 when BT crops were not yet widely adopted, through 2017 when 90% of corn and cotton acreage was planted to BT crops. Individuals were randomly selected from collections made in these 2 years, and we sequenced their genomes. With these data, changes in the frequencies of alleles over time can be quantified to identify resistance-related genomic changes. So these were the results from our temporal genome scan. On the x-axis, again, you have chromosome number, and the y-axis gives our measure of allele frequency change or divergence, and here we're using FST. Each point essentially represents allele frequency divergence between populations collected in 2002 and 2017 in a single 10 kb genomic window. Points above the red dashed line represent regions of statistically significant genomic divergence. And points in color represent regions of strong divergence over time that overlap with QTL regions. And what's notable about this figure is that the blue peak in chromosome 9 shows the second greatest genomic divergence over time, and it overlaps with our Cry1AB resistance QTL. We also examined coverage depth in samples collected from these same sites in the years 2002, when there was no documented Cry1AB resistance. 2012, after Cry1AB resistance warning signs had emerged in field Zea, and 2017, when resistance to Cry1AB was causing field failures. What we can see from this series of panels is that coverage depth increased over time, indicating that copy number variation in this region increased as resistance grew in wild Helicobacter. So copy number variants are important to rapid evolution, and these, along with point mutations, produce much evolutionary novelty. In fact, our gene copy number amplification, or rather gene copy number amplification in general, is a well-described mechanism of pesticide resistance. So perhaps we should not have been surprised to have found it in our study. However, ours is the first case of a copy number variant that we know of associated with field-evolved BT resistance. So just to conclude, Helicobacter resistance to Cry1A toxins, in our work Cry1AB, but recent studies have also shown this is the case for Cry1AC. Resistance to Cry1A toxins is polygenic. And in our case for Cry1AB, additional QTL of moderate effect size will require further investigation to understand their function and role in resistance and the types of mutations that led to resistance evolution. Polygenic resistance likely resulted from Zea's low-dose exposure to BT toxins in agroecosystems, and this supported theoretical predictions. And copy number variation appears to play a substantial role in Helicobacter's rapid adaptation to transgenic BT crops. To our knowledge, this is the first copy number variant implicated in high levels of resistance to any transgenic crop. And just to leave you with some future directions for our lab, these are the genes in the copy number variant region of chromosome 9. And what you might notice from this chart is that the majority of them were previously annotated as trypsins. Initially, we were excited about this. Trypsins were previously known to function in BT resistance. In general, trypsins are enzymes that are secreted into the midgut, and their job is to cleave proteins. They have general digestive or metabolic function. But they're also known to play a role in activating cryprotoxins. When they cleave at a specific amino acid, it activates the crytoxin, allowing it to bind with an interacting partner protein or receptor along the midgut wall. Past studies had shown that mutant trypsins can no longer activate cryprotoxins, and this resulted in a loss of receptor binding and a loss of toxicity. But our mutation appeared to be a gain of function mutation, increasing the copy numbers of a gene, which increases gene expression, not a loss of function mutation. Moreover, many cultivars of Cry1AB expressing corn are already expressing activated toxins, not the protoxins, so that any mutations involved in reducing toxin cleavage should not be a field-relevant mechanism of resistance in Zeah. So, given the general digestive function of trypsins, we also thought that perhaps the excess of expressed trypsins resulted in rapid degradation of crytoxins in the midgut, as had been proposed for some other heliothyme species. These degraded toxins should not be able to bind to receptors, resulting in a loss of toxicity. For this to be the case, these trypsins should have what is known as the catalytic triad, amino acid residues of the trypsin protein that work together to facilitate the hydrolysis of the toxin's peptide bonds. However, all of the trypsins in this region lacked the catalytic triad. The red amino acids point to where there has been a substitution in the active site. And this indicates that these genes may not have proteolytic function at all and should instead be considered serine protease homologs. When we modeled the structure of the proteins using AlphaFold 2, we could see that these serine protease homologs have still retained a binding cleft, despite lacking the catalytic triad. So we're currently working with collaborators at the University of Tennessee, Knoxville, to test these genes for their potential functional roles in resistance. So with that, I'd like to acknowledge the amazing work of the trainees in my lab, Dr. Kate Taylor and Jane Quackenbush. I'd like to thank the USDA for the support over many years of my research program and the many collaborators who have been instrumental for us in doing this work. And with that, I'd invite you to email any questions to me here at my University of Maryland email address. And I appreciate the opportunity to share my work with you today.
All right, thank you, Megan. Yeah, I see someone clapping there. Really, really cool talk. I had a question, and someone else in the chat had a similar question. So when you were showing us that there was QTL responses for the two different treatments, right? One toxin versus two toxin. You focused on chromosome 9, and I noticed that for the two toxin treatment, that peak was also there. Have you looked to see if it's the same genes driving that QTL in both treatments?
That's a really good question. So we know that the gene amplification event is very common in across Helicoverpa Zeah in the landscape right now. You know, if you go out and take a random sample in a lot of locations, something like 90% of individuals will have a copy number variant in that region. And so we expect that because the two toxin treatment that we were looking at is Cry1A 0.105. It's a Cry1A toxin, and that probably the gene amplification event was there in the grandparents we used for those crosses, and that probably is underlying to some extent resistance to the two toxin cultivars as well. But we suspect that there are probably other things on that chromosome that are also involved in resistance to those two toxins because of the elevated additive effects across the entire chromosome. So.
Okay, cool. And kind of along that same note, you already answered this in the Q&A, but when you're looking at field resistance, there was more than one peak of things that looked like there was one on chromosome 13. And I know you've already addressed it, but if you would just say it out loud as well, like what's going on with that big one?
Sure. So we did look at that in our first paper. Actually, we got really excited when we first saw this peak because we thought, oh, maybe this is the thing that's underlying cry resistance in Zea. But when we actually overlaid the results of our QTL analysis, we found no evidence whatsoever that chromosome 13 is involved in any way in resistance in the populations we looked at in response to any of the cry toxins. So what is under that peak is it's actually a very narrow peak, and there's just like a handful of genes, and the three that really stood out were a cytochrome P450, 333B3, as well as two carboxypeptidase Q genes, or they were annotated as carboxypeptidase Q. So we sort of hypothesized that those genes were probably under strong selection by other things in the environment, and it could be, given a cytochrome P450 and that Zea are exposed to pesticides, it could be selection by synthetic pesticides in the environment, like pyrethroids, or that it could be secondary plant defensive compounds that they may be exposed to in the environment. But not associated with BT resistance, unfortunately.
Cool, thank you. And then Brenda, I think you wanted to ask a question. You can go ahead.
Of course. Hi, Megan.
Hi, Brenda.
We talked a little bit about that you were presented some of this work at PAG. I don't think you presented the end of what you're working on right now on these serine protease homologs. And that is intriguing to me because that looks very similar to what we see in protease-mediated resistance, where we have these serine protease homologs that hang around. I haven't looked to see that if they're duplicated. That's fascinating that they would be duplicating maybe a homolog. And what we have proposed is that they serve as sequestration of the inhibitors. And so I'm wondering if there's a way, you're working with Juan Luis on this, right?
Yes, I thought I saw Juan Luis in the audience too. So,
okay, wondering if there's a way you can quantify the amount of toxin, maybe do a comparison like that to see if actually they are removing, the activated toxin from the gut.
Sure. So this is not my area of expertise, which is why I'm very grateful for the collaboration of Juan Luis. And yeah, so it is something that we intend to do, and we're working on getting up and going right now with this. But one of the things that is really interesting to me is that it seems like there's selection for, like, we can see within a single generation selection for higher copy number. And so if we go out in the field and we collect on pairs of plots, BT expressing and non-expressing plots, we can actually see that you can still collect a handful of susceptible individuals in non-BT plots, but you can't collect any single copied individuals off of corn that's BT expressing. And so that would suggest that there's something important about having many copies of this, and that perhaps they are doing the job of sequestering BT toxins.
Is it possible that they are also responding to protease inhibitors in the corn plant? And so it's just coincidence that they're expanding this that happens to also be beneficial to avoiding the toxin?
I have two lines of evidence that I don't think that that's the case. The first is that when we do all of our experiments in the lab, we have a control, and it's still corn tissue. It's the non-expressing isoline, right? And so if it were really just a response to the corn plant itself, we wouldn't expect to see this QTL only in the BT expressing treatment, right? And then also when we can still collect plenty of, you know, there are very few that seem to be out in the environment, but we can still collect the single copy individual off of a non-BT isoline of corn in the environment, but we just can't do that on our BT expressing corn. And so I don't know if that helps to address your question, but I don't think that that's the case. That would those lines of evidence would seem to suggest that it's really related to the toxin itself.
And so there is still planting those events in the field right now?
They do. Yes, that's right.
And what are the measures that they're using to combat resistance then?
So that's a really good question. I think in general, I'm not an expert in management of Zeah in corn, but what I'll say is that management in corn Zeah infestation seem to not be a huge deal for corn producers. One of the things I will say is that they can stack into those cultivars or pyramid into those cultivars VIP3A, which still retains activity against Zeah. And so that's also really important and critical because it also protects cotton. And I think cotton damage by Zeah is much more economically important than in corn production. And so having VIP3A in the field and planted with pyramided into corn cultivars that are transgenic is still important, and it is managing Zeah at this point.
Okay, thank you.
And then Juan Luis was in the attendees, and I made it so he can talk if he feels like it, but we won't put him on the spot unless he has something he wants to say. All right, just a couple more questions then before it's the top of the hour. Greg Sword says, great talk. What is indicated by the strong negative additive effects on chromosome 10?
That's a good question, and one we've thought a little bit about. We actually think that there might be something going on there related to the diet that we use, the lab diet that we incorporate the leaf tissue into. And so with the y-axis is the additive effect of having a resistant parent allele, but the alternative would be if you had a susceptible parent allele, that perhaps there's something that benefits you from having that susceptible parent allele given our treatment. And so we've been thinking that probably that's related to the fact that we do mix corn tissue in with a lab diet for us to do this assay, and everybody else does, uses lab diet as well for these assays, just overlays instead of leaf tissue incorporation. So that's, I think, the best I can do to explain that part.
Thank you. And then our very last question is just one out of curiosity from someone outside of the field. For pest control, if transgenic antimicrobial peptides or other toxins are expressed, can we expect the insect or pest to develop resistance through increased expression of trypsins or serine proteases?
So I'm sorry, is the question for other?
I think so. It's just kind of a general question from a curious person here.
It is a good question. And I guess I'm not entirely certain of the answer to that question. It could be possible. But I think that there are other mechanisms that may play a role as well. And so one of the things that I've talked to colleagues about is the fact that there are many cases of field evolved resistance where instead of it being these trypsins, there are receptors or genes that encode receptors for these plant incorporated protective transgenes, or the proteins that they manufacture, that are important to resistance. And so there are probably more, there are more cases of a resistance mutation in a receptor than there are in, at least to date, in field evolved resistance and by this mechanism. So.
Okay, thank you. Well, thank you, Megan, for a great talk and answering all the questions. Thank you to all the speakers today. Everyone did a great job, and thank you to the attendees for asking questions. It's always more interesting when we have things to discuss at the end. So we made it to the top of the hour. I'm not going to hold anyone away from lunch. Can't stand in the way of food. So I just want to thank the organization team and everyone for allowing us to do AGSx one more year. So with that, I'll sign off, and everyone have a great rest of your day.